Welcome to this explainer. You know, if you've ever stood in the produce aisle looking at perfectly uniform, pristine fruits and vegetables and wondered exactly how agricultural science is keeping pace with a booming global population, well, you are in the right place. Today, we're unpacking something truly wild. We're going to look at how cellular manipulation and tissue culture act as literal superpowers for modern scientists, letting breeders essentially engineer the very future of our food. Here's our blueprint for today. We'll be covering the Modern Breeder's Toolkit, the Ploidy Matrix Explained, the Time Machine Shortcut, engineering trades with triploids, breaking natural plant incompatibilities, and finally, the integrated breeding pipeline. Let's dive right in.
Starting off with Section 1, the Modern Breeder's Toolkit, focusing on the tissue culture ecosystem. The massive world of tissue culture is basically broken down into four distinct functional quadrants. You've got trait engineering and genetics, stress selection, mass production, and conservation. Together, these form the absolute bedrock of advanced crop improvement. But for our purposes today, we're going to focus heavily on that first one, trait engineering and genetics. Why? Because that is where breeders actually get in there and alter the genetic destiny of a plant.
Moving right along to Section 2, the Ploidy Matrix Explained. Let's talk about genetic states.
So a plant's chromosomal state completely dictates its ultimate agricultural potential. Think of it like this. Haploids, which have just one single set of chromosomes, allow us to totally expose hidden recessive traits. Then you have double haploids, which give us instant 100% pure genetic lines. And triploids, well, those yield the seedless, physically massive crops we all love. Each of these states plays a hugely crucial role in the modern breeding pipeline. All right, Section 3, the Time Machine Shortcut, where we look at accelerating homozygosity.
Historically, creating a pure, highly stable plant line required a ridiculous amount of patience. We're talking 7 to 8 years of continuous self-pollination just to weed out genetic variability. But the double haploid shortcut? It's a literal cheat code. It collapses that entire decade of painstaking, repetitive work into a single year. So instead of waiting generation after generation, slowly weeding out the genetics you don't want, breeders bring in a chemical called colchicine. This incredible tool transforms an initial mixed-up hybrid directly into a pure, homozygous line in just one generation. You get the exact same perfectly pure result. You just saved, 7 years of waiting around. Now, pulling off that incredible shortcut does require some serious high-tech lab control. In the first phase, scientists take reproductive tissues, like anthers, for example, and plate them in a culture. By essentially tricking these gametes, they force them to develop directly into embryos. This leaves us with a fragile, intermediate haploid plantlet that only has half of its normal chromosomes. Then, in phase 2, we take that delicate little plantlet and deliberately double those chromosomes. By triggering this doubling with colchicine or even highly precise CRISPR-Cas9 techniques, that half set of chromosomes perfectly duplicates itself. It instantly locks in absolute genetic purity, and boom, we have our stable double haploid line. On to Section 4, engineering traits with triploids. Let's see how we direct plant energy. I want you to think about this for a second. If you've ever bought a seedless watermelon, you've probably wondered at some point, how on earth can a plant with no seeds reproduce? Like, how is it even grown commercially in the first place? When you stop and think about it, it feels like a total biological paradox. The secret here is triploid math. Breeders strategically cross a 4x female plant with a standard 2x male. The resulting offspring is a 3x triploid. And since 3 is an odd number, those chromosomes can't pair up evenly, which makes the plant completely sterile. But here's why that's brilliant for us. Without wasting energy developing hard, annoying seeds to spit out, the plant redirects all of its metabolic power into growing much larger, significantly sweeter fruit. And tissue culture pushes this concept even further. We can actually completely bypass that whole complex cross-pollination step entirely. By taking naturally triploid primary endosperm and culturing it in a petri dish, scientists can induce callus formation directly. So we're going straight from endosperm to a fully formed 3x plantlet, all within a highly controlled lab setting. Section 5, breaking natural plant incompatibilities. Time to meet the barrier breaker. Out in nature, plants have evolved some pretty sophisticated defense mechanisms. When pollen lands on a stigma, it has to grow a tiny tube all the way down the style just to reach the ovary. But plants actually have physical and chemical walls right there that actively reject pollen from different species, or sometimes even from the plant itself. This strictly limits what traditional breeders can pull off out in the field.
So how do we get around a biological fortress like that? By using in vitro fertilization. Scientists can completely bypass those natural barriers by fertilizing the ovules directly in a test tube. This enables literally impossible crosses between totally different species. It allows for fertilization within self-incompatible lines, triggers haploid production, and crucially, it lets us introduce vital, climate-resilient traits like severe heat stress tolerance into staple crops like maize. It's an absolute game changer for protecting our global food supply. Which brings us to our final section, Section 6, the integrated breeding pipeline. This is the masterpiece. This is where the compounding power of tissue culture becomes truly unmatched. A breeder starts by using the barrier breaker, that in vitro placental pollination we just talked about, to force a completely impossible cross between two different species. The result is a highly novel but totally sterile and unstable hybrid. But then, they immediately bring in Tool 1, the time machine. By taking the anthers from that wild hybrid, culturing them, and applying colchicine doubling, they instantly stabilize that novel genetic mix into a perfect, elite stable line, something that would have been biologically impossible in nature and taken decades to refine even if it were possible, is fully accomplished in a matter of months.
Seeing how all these incredible tools compound, how breeders can just seamlessly bypass nature's limits, isolate the absolute perfect traits, and accelerate decades of evolution into a single year, it really makes you wonder. We have effectively rewritten the rules of biological possibility. So the question I'll leave you with today is, just how radically will we be able to shape the future of our food? Thanks for joining me for this explainer, and I'll catch you next time.